Good morning, everyone. On behalf of Indo-Pacific Future Leaders in AMR, it's my great pleasure to welcome our speakers and participants to our inaugural online student symposium. Before we begin today's event, I'd like to acknowledge the traditional owners of the country. Can I have slide 2, please? Yeah, we also encourage our participants joining from outside Australia to honor and acknowledge the Indigenous people and cultures from the lands from which you join us today. We acknowledge and pay our respects to the people of the Kulin Nation and all across the world. We recognize the owners and traditional custodians and recognize their continuing connection to land, water, and culture. We honor elders present and past, whose knowledge and wisdom has ensured the continuation of cultural and traditional practices. We are committed to genuinely partner and meaningfully engage with Australia's traditional owners, Aboriginal and Torres Strait Islander communities. Now, today's symposium has been organized by a symposium organizing committee as part of the Indo-Pacific Future Leaders in AMR. It is a tremendous job to put together an international event from scratch, especially for the first time. The conference has brought together over 100 participants registered from 8 countries, 17 research institutes, and 34 centers for learning and discovery. The main goal of this symposium is to foster networking among the early career researchers in the Indo-Pacific region. While we look forward to hearing about some exciting research about one of the key global health issues, we also hope that you, as future leaders, would enjoy connecting with passionate AMR researchers in the symposium across different countries. Thanks again, everyone, for joining us today. Just a gentle reminder for participants to stay on mute during the talks, unless you're presenting. For questions related to the talks, please submit them to the chairs during each session using the Q&A function or the chat box. We also have a Google couple of prizes for the best questions, so we'd encourage you to actively participate in the discussions. So now, without further ado, I would like to invite Professor Anton Peleg, Director of Monash Center to Impact AMR, to please say a few words about our center. Thank you.
Thank you very much. And, this is such an exciting day. I know there's been just a huge amount of work that has gone in from the organizing committee, and it's really just an inspiring initiative. So, we're all here for a reason to really think about this global problem of antimicrobial resistance. There's clearly been some really important literature come out to suggest the sheer impact of antimicrobial resistance and the burden on mortality globally. And recent data would suggest that there's just under 5 million people that are currently dying each year of a bacterial AMR infection. And unfortunately, a lot of this burden is predominated within the low and middle income countries, and particularly our region in Asia. So I think this initiative is just so crucial for developing new solutions, thinking in an interdisciplinary way, as well as bringing our geographic neighbors all together to think in a multidisciplinary way. I think this is really, really exciting. I think from the Centre to Impact AMR, this is amazing to be able to support such a cross-country initiative. And to have everyone within the region to contribute, and particularly to have this come from the students and the junior group, who are really going to be, as the title of today's symposium is, the future leaders of AMR. The centre, its goal is and vision has been not only to stop the progression and surge of AMR, but also to reverse it using novel solutions and sustainable solutions. And I think today is one of those symposia where it can bring amazing young minds with brilliant ideas together. So I encourage all of the speakers to really use today as a great platform to discuss and think about new ideas. I wish the day extremely well. I know you have some fantastic speakers, and it looks like a great symposium. And I really support this with my greatest enthusiasm. So have an amazing day. Well done to the organising committee, and enjoy and immerse yourself and open your mind to novel solutions. Thank you.
Thank you very much, Anton, for that very moving speech. So to kick off the day, it is our first keynote address. It is my great pleasure to welcome Professor Mark Blaskovich to the symposium. Professor Mark Blaskovich is an antibiotic hunter and director of the Translation for the Institute of Molecular Bioscience at the University of Queensland. He's also the director of the ARC Industrial Transformation Training Center and the Antibiotic Crowdsourcing Initiative COAD. He's a medicinal chemist with 15 years of industrial drug development experience at 3 biotech companies. And since 2010, he's been developing new antibiotics, non-antibiotic therapies, and diagnostics to detect and treat resistant bacterial and fungal infections, including having multiple industry collaborations focused on antimicrobial resistance. Mark's talk today is titled, How Do We Combat AMR? Global Collaborations for a Global Problem. The floor is yours, Mark. Share your slides whenever you are ready, and we'll go
That
from
would be
there.
great. All right, hopefully everyone can see that in presentation mode. Excellent. All right, well, thank you very much for the invitation to present. I really appreciate the opportunity to talk to the next generation of antimicrobial researchers. And as Anton says, you guys are really important, and that'll get highlighted a few slides later. We're showing that we really do lack new researchers entering into the field of antibiotic discovery, development, and antimicrobial resistance. So you guys are key to finding solutions to combating this global problem. So I thought I'd give a bit of a perspective from an antibiotic discovery side. So I think a lot of people are very familiar with antimicrobial resistance and where it's come from and where it arises. But I think some of the problems about developing new antibiotics are probably less known by a lot of people. So I'm going to focus my talk on that. I too would like to acknowledge the traditional owners, particularly the Jagera and Turrbal people whose land on which we conduct our research. And I'll start out just by giving an overview of antibiotic resistance and the development of antibiotic resistance. So this is a slightly different perspective that came out in a paper a couple of years ago, which looks at the fact that, so initially when penicillin was first discovered, resistance to penicillin was identified even before it was used clinically in patients. But for every antibiotic since, the antibiotic was introduced, and anywhere from a couple of years to a couple of decades later, significant resistance was identified in the population. So antimicrobial resistance is nothing new. Every antibiotic introduced, resistance has developed. And this is what this paper called the established timeline. But since about 2005, what they've now found is that predominantly because they do a lot better testing of the compound before it makes it into a final approved drug, they actually identify the resistance a long time before it's introduced clinically. So no longer do we have this gap between the clinical use and the identification of resistance. We actually have the resistance identified well before the antibiotic starts to be used clinically. And so, this is obviously of concern as we introduce new antibiotics. They're essentially already to some extent obsolete. And it raises a question, so if resistance to antibiotics has been around for so long, why is it becoming such an issue now? And it really comes down to the lack of development of new antibiotics. So this slide's a bit old, it's from Carbex and from about 2018, but it shows that you had this golden era of antibiotic discovery. Golden age of antibiotic discovery between the 1940s and 1980s, where basically all the classes of antibiotics that we now use clinically were discovered. And since then, there's been a dearth of approvals of new antibiotics representing new classes. Now, this figure is slightly nuanced in the terms of we have discovered new classes of antibiotics, but they have not yet been approved clinically, or we have identified clinically approved new classes of antibiotics, but they were first discovered prior to the 1980s, like the first member of the class. So when you see this picture, there are some nuances in how it's actually identified. But it also correlates. There's a report came out called Leaving the Lab last year, which identifies the FDA approval of antibiotics. And again, it matches, since 1985, the sharp decline in the number of approvals of antibiotics that get approved every year. And for the ones that are approved, they're pretty much all new and improved versions of existing antibiotic classes. So we're not developing new classes of antibiotics, which means they're generally susceptible to the same mechanisms of resistance, although some of the new and improved versions have been designed to overcome some of those resistance mechanisms. But what this means is that if you look at the number of drugs in clinical trials and you compare antibiotics, which are one of the few medicines that actually cures the disease and regularly saves millions of lives annually, you compare it to oncology drugs. Within the 3 stages of clinical trials, so you have phase 1, phase 2, and phase 3, there are approximately anywhere from 1,500 to 1,800 oncology drugs currently in development in those clinical trials. And you compare that to antibiotics, there may be 50 or 60. And so this just highlights what a difference in the investment of research is going on into antibiotics, which are arguably one of the best medicines ever invented, compared to other types of therapeutics. And part of the reason for that is that it's very hard to discover new drugs. And so I'm not sure if you're familiar with the drug pipeline, but basically it takes a long time and there's a lot of attrition of potentially new compounds along the way. So in that early stage discovery, you've got literally thousands of potential compounds, and those get winnowed down into the ones that enter formal preclinical development. And then you have this handful of compounds that make it into human clinical testing, the first stage of phase 1, which is looking at basically at safety. About half of those fail, so now you're down to half of those making it into phase 2, which is looking at initial studies of do they actually work at curing the disease. And then about half of those fail, and so now you're down to a couple of candidates left in phase 3 in the big large-scale clinical trials and thousands of people. And then approximately half of those fail. So by the time you get to the ones that are actually approved and registered and marketed as a new drug, you're talking about anywhere from 10 to 15 years if you're lucky, and an overall cost of close to $1 billion, if not more. And so it's very, very expensive and takes a lot of time. And this is why there's this urgency. So the predictions are by 2050, there'll be 10 million deaths per year caused by antimicrobial resistance. And the reason that's so critical is that if we don't start acting now, it's almost 20 years before we can develop a new antibiotic. And so we have to be putting a lot of effort into early stage discovery now to start priming this pipeline so it can end up with drugs, in 15 to 20 years' time. So why is there such a lack of research into antibiotics? And it's largely economic reasons. So if you look at what you can charge for the best new approved antibiotic that's on the market today, is approximately $1,000 a day. So if you're looking at your standard 2-week treatment, you're saying about $14,000 to $15,000 for reimbursement for a new antibiotic. And you compare that again to the newest anti-cancer therapy, so CAR T therapy, is about close to $500,000 per patient. And so if you're a drug discovery company and looking where to make your investment, you're going to put your money, given it costs about the same amount of money to develop one drug versus the other, you'll put your money into the one that makes you $500,000 per patient versus the one that you can barely break even for your cost of goods. And this is reflected if you look at the new drug sales. This is from about 2018, so it's a while ago. But 2 of the most successful new antibiotics approved were Teflaro and Avicaz. And each of those, for the first 2 years of sales after approval, they made about $50 to $80 million each, which sounds like a reasonable amount of money. But you compare it to Lyrica, which is a drug for epilepsy, neuropathic pain, or Januvia, a diabetes drug. And both of those made over $1 billion in their first 2 years of sales. So again, if you're an executive in a company, why would you be investing in antibiotics when you can't make any money out of them? And so this is the main reason why pretty much all big pharma has left antibiotic discovery and development. So Novartis shut down its efforts. Glaxo is still doing some work, but has largely shut down its early stage R&D. Pfizer supposedly moved a lot of its research to China, but there doesn't actually seem to be much research going on there. Sanofi quit in 2018 as well. So there's been this large exodus, and prior to 2018, most of the other companies had already left. AstraZeneca pulled out in 2016, and when AstraZeneca pulled out, it kind of highlighted where the shift in antimicrobial research went, because they put all their antibiotic assets into a company called Entasis, which was a startup spin-out company. But I'll show in a second, the reliance on the small biotech companies for drug discovery isn't going very well. And for example, Entasis a couple of years ago was doing very poorly, and it was acquired by basically a money-making company called Inovia, which took over the Entasis asset that was closest to approval and basically sidelined all their fundamental research. So if you look at biotech companies, there are a number of excellent case studies which illustrate the problem with relying on biotechs for antibiotic discovery. Probably one of the best known is a company called Keynote. So it had a new antibiotic, aminoglycoside called plasmamycin, that got approved in 2018. So for most biotech companies, if you get a new drug approved, your stock price starts shooting up because now you've got something on the market and you can make money. For Keynote, what happened is their stock went into a steep decline right after approval, and they had to file for bankruptcy less than a year later. And part of that is the ongoing costs of after you get a new drug approved. So their R&D expenses were about 103 million for 2018, the year when it was approved and a bit after. The maximum cost they could get per patient was $2,700 US. And so their revenue for the first year of product approval was less than $1 million. And the amount of money they had invested in the company to develop that drug was over the years close to $1 billion. And so the revenue they were getting was not even covering the costs of manufacturing and producing the drug, let alone all the money that had been invested into developing the drug. A similar story with a company called Tetraphase, which was developing new tetracycline antibiotics. Their stock price actually went down well before they had their market approval, but post-market approval, again, the stock price went even lower. 300,000 in net product sales for the first 3 months, and an accumulated deficit of $500 million, despite contributions of over 300 million from BARDA. And so Tetraphase has also gone out of business. A company called Melinta Therapeutics had a new quinolone approved in 2017. First year sales were 6 million. Their total incurred losses since the founding of the company was nearly $750 million. So all of these examples, including another one, Paratech, which shows their antibiotic approved in 2018, and their stock price also declined. So all these companies are excellent examples that, relying on small biotechs to develop new antibiotics is not feasible under the current financial climate. And this is why, venture capitals that invest in the small biotechs look at the results from these companies and won't invest into antibiotic discovery companies. And so this figure highlights nicely all the different small biotech companies that were focused on antibiotic discovery development and what's happened to them. And so basically, they're all out of business now. Their antibiotic assets, some of them are still being developed or marketed by another company. Other assets have just disappeared and are no longer being developed. There's a investment firm that's focused on antibiotic called the Novo Repair Fund, and supported by all the money that Novo Nordisk makes out of particularly their diabetes and weight loss drugs. And they put together a little overview of the financing that goes into oncology versus antibiotics and antifungals. So this figure looks at the annual money raised in US dollars for oncology companies versus antibiotics and antifungals. And so there's 42-fold higher amounts, so 50 billion invested into oncology companies versus 1 billion in the antimicrobial. And the number of deals made for acquiring the company or acquiring the assets, nearly 1,700 versus 102. So again, all the money is going into other therapeutic indications. There's very little going into antibiotic discovery development. And this has led to the other crisis, which is basically the exit of antimicrobial researchers from doing antibiotic discovery and development. And so this paper tracked the number of AMR publications for the last 30 years, and there's been the sharp decline in papers focused on antibiotics and antifungals. And again, comparing it to, for example, cancer, there's been a steady increase, whereas you have this decline in antimicrobials. And they looked at researchers that had left antibiotic-focused companies. So this was 6 firms, 150 people that used to work on antibiotic discovery development. The next company they went to, only about a quarter or a fifth of them were in a company focused on antimicrobials. And when they did another shift now, you're down to about 10%. And so this highlights the exit of all this experience in antibiotic discovery and development from industry into other therapeutic areas. And so we're basically losing all the collective knowledge we used to have. Estimates are back in the 80s, there are probably 20,000 researchers in industry focused on antibiotic discovery and development. And developing a new antibiotic has certain, there are certain peculiarities to drug discovery of an antibiotic versus other types of therapeutics. And so that collective knowledge and wisdom is now being lost. And so these people in a biotech company that are developing new antibiotic are having to go through all the same trials and tribulations that were already, the solutions were figured out 40 years ago, but now no one remembers what they were. And so they estimate, looking at a variety of summaries, that there are approximately 3,000 antimicrobial researchers left globally. And so this is why you guys are so important. You know, the number of people that are on this call, if you're going to continue in your career in antimicrobial resistance, you make up a pretty significant percentage now of that 3,000 researchers left in antimicrobial resistance. So what are the solutions? So obviously, one of the big problems in antibiotic discovery development is a lack of money. And so we need funding incentives. So there are 2 different types of funding incentives. I'm really dating myself here with this picture. This is a Dr. Dolittle slide, and there's an animal in Dr. Dolittle called the push me, pull me. So push incentives are things that are money is pumped in to fund antibiotic discovery development. So this is a kind of traditional funding that within Australia, you know, NHMRC, ARC grants. MRFF, I put a question mark behind that because even though technically there's meant to be a bunch of MRFF money going into antibiotic discovery development, there's actually very little. Most of it's going into surveillance and clinical related activities. Therapeutic Innovations Australia has voucher programs that allow you to access some antimicrobial resistance. The Cummings Centre has had some antimicrobial calls as well. And then there are a number of global antimicrobial specific schemes where Australians have access to. The most significant of them is a company called CARB-X, or an entity called CARB-X, which is a not-for-profit with close to $1 billion that was contributed by the Wellcome Trust, the US BARDA agency, and several other countries that is specifically devoted to advancing early stage antibiotic assets into phase 1 clinical trials. And then there are several other initiatives there, JPIMR, the PACE in the UK, NIH, NIAID, and BARDA are US entities that have had significant efforts focused on antibiotic discovery. In the current climate, I suspect getting funding out of them was going to be very difficult with the double whammy of the new head not really believing in infectious diseases, plus the lack of appetite for funding foreign entities. And then later stage, the AMR Action Fund and Novo Repair Fund are funding companies that have antibiotics in clinical trials. The other side are pull incentives where you're looking to reward a successful research outcome. So for a company that is able to get an antibiotic approved, rather than relying on sales to generate their revenue, the idea is to guarantee them a significant financial reward for getting that antibiotic approval. And so one of the most interesting approaches that's being trialed is something called the Netflix subscription model. And the idea behind this is that if you have an antibiotic on the market, a country will guarantee you a fixed revenue per year for having access to that antibiotic rather than paying you when they use it. And so the UK has been the pioneer of this. They implemented it on a trial basis for a couple of antibiotics, and have now rolled it out into a full system. The problem with the Netflix model is that you basically need most countries to subscribe to the model in order to make the global financial reward sufficient. So the UK is basing their reimbursement on their share of the global antibiotic market. Australia is considering it. It's being proposed by several entities. The biggest market is the US, and there's a act before Congress called the Pasteur Act, which has been sitting there for close to a decade and still has not been approved, which would fund a similar outcome. And that would be a game changer, because that would provide a big chunk of money that would give confidence to antibiotic discovery that if they get an antibiotic approved, there is a pathway for them to make significant revenue. The other ideas are things like market vouchers. So this is if you get an antibiotic approved, you're awarded a patent life extension on some other drug within your portfolio. So if you've got a oncology drug that's making $1 billion a year, you could get an extra year of life before it becomes generic, and so you get an extra $1 billion in revenue. And if, for example, you're a small biotech company that's just focused on antibiotics, you get an antibiotic approved, you can trade that voucher to a big company that has one of these much more lucrative drugs on the market. And so that's a way of essentially giving $1 billion, but indirectly through funding from drug payments. There also is a market reward. So the AMR Action Fund is a fund of close to $2 billion. The idea is if an antibiotic is approved and meets certain criteria for being able to treat resistant infections, there's this pot of money of up to $1 billion that can be awarded to the company that had that antibiotic approved. But you notice I'm talking about big chunks of money, right? It's not a little bit of money. We're talking about billions of dollars. So are these incentives working? So CARB-X started in 2016. Some of these other initiatives have come along since then. We've been tracking the number of drugs in the antibiotic pipeline since 2011. So we've got over a decade looking at how many antibiotics are in those 3 different stages of clinical trials and how they're progressing. And so if you look at the number of drugs in phase, I'm going from left to right, phase 3, phase 2, and phase 1, back in 2011, you see this kind of inverted pipeline. So normally what happens, you got a small number of drugs in phase 3, about twice as many in phase 2, and then you should have about twice as many in phase 2 in phase 1. In this case, going from phase 3 to phase 2 is about what you'd expect to see, but this number of compounds in phase 1 is less than the number of phase 2. And so this is where concerns about the antibiotic pipeline started to come from, because if you only have 12 candidates in phase 1, another 5 years, you'll have half of those in phase 2, and another 5 years, half of those in phase 3. So the diminishing returns means, this is what the number of potential compounds that will be approved in 15 years' time. And so it's very, very low. So as you go over the years, things have actually improved. So we've now got more compounds in phase 3. We've got a lot more compounds in phase 2, and we almost have as many compounds in phase 1 as in phase 2. And so it does seem like these efforts to stimulate the pipeline are having an effect. We do have more promising new candidates starting to go into the clinical pipeline. So it's promising, but it's still not enough. Like overall, we're still talking about 60 compounds in clinical trials versus the thousands in other types of indications. So moving on now to the research side, how do we actually go about discovering new antibiotics? So the approach that has been most successful for the past 4 decades basically is to rationally approve existing classes of antibiotics. So you take an existing antibiotic, try to make it more potent, less toxic, and overcome existing resistance mechanisms. And this is why we have 5th generation beta-lactams, 3rd generation tetracyclines, et cetera, because there's been it's comparatively easy to modify something already generally works. There has been some effort to rediscover old antibiotics. So I mentioned that golden age of antibiotic discovery. They were in the 50s, 60s, 70s. There were literally thousands of new antibiotics reported in the literature. Only some of those were advanced into clinical trials, partly because we had so many options at the time. So can we go back and find some hidden nuggets within the literature that have promise but weren't developed at the time. There has been some effort to look at repurposing old drugs. So drugs approved for other indications, some of them have been found to have antimicrobial activity. Not a lot of success there. Possibly one way of using them is using them as in combination with existing antibiotics. There's been some drugs that have been shown to act as potentiators. Target-based approaches, so bacteria have very different genetic makeup than human cells. So theoretically, it should be possible to find a lot of targets that are unique to bacteria. And this was an approach that was focused on heavily in the 1980s and 90s without a whole lot of success. And so what happened was you have a target-based approach, you isolate a target, identify a target that's unique to bacteria, you find a compound that, for example, if it's an enzyme, able to inhibit that enzyme with exquisite potency, then you go try treating the whole cell of the bacteria, and the bacteria have all these mechanisms to keep stuff out of them. And so even though your compound is very potent on your target, it actually can't get to the target because the bacteria either efflux it out or have membranes that stop it from getting in. And so most of these approaches have been unsuccessful. Some of the targets that were discovered and initially worked on in the 1980s, they're finally starting to get success now, 30 years later, because they're able to start identifying ways to overcome the efflux and entry difficulties. So we do have some compounds entering the pipeline now which are focused on new targets, but it's not easy to do. And then you can go back to how antibiotics were originally discovered and look at untested new chemical diversity. And so almost every antibiotic was originally discovered as a natural product. And most of them were discovered from soil bacteria or fungi. And so there's been a renaissance in natural product discovery. It fell away for a while because after the golden age, people just kept rediscovering the same antibiotics because they were using the same type of technology. But now with metagenomics, whole genome sequencing, the ability to try different culture conditions to grow up the bacteria which don't grow under standard conditions, we are identifying quite a few novel antibiotics from natural products again. But the approach that we want to look at was tapping into synthetic chemical diversity. And so this is what we set up our community for open antimicrobial drug discovery to do, to try seeing if we can find unusual chemical diversity from the millions of compounds that synthetic chemists have made over the years. And so we set up COED as basically an academic crowdsourcing initiative trying to discover new antibiotics. And our hypothesis was based on the fact that antibiotics are not drug-like, right? So for drug discovery, there are a bunch of rules that are now in place to try predicting, in particular, whether a compound is likely to have oral availability. And Big Pharma is curating all their compound collections based on these rules. They want to give themselves a higher success rate of having something that's likely to be drug-like. But if you look at approved antibiotics, which were, again, mainly originating from natural products, most of them break a significant number of, for example, the rule of 5. They also often have these reactive functionalities. And so, again, Big Pharma uses filters to get out reactive functional groups because they generally don't make good drugs. But if you applied all these rules, you wouldn't have all these different classes of antibiotics. They never would make it through a modern medicinal chemistry program. And so if you look at where compound collections are now are clustered, if you look at 2 properties, molecular weight and lipophilicity, complying with rule of 5, this is where biotech libraries and commercially sourced libraries sit in chemical space. But if you look at where academic antibiotics, so these are compounds reported in the literature that have antimicrobial activity, much greater chemical diversity. So if you're only looking in this space for a new antibiotic, you're potentially missing out all this other space around. And this is kind of where COED is trying to tap into these academic structures that have fall outside the traditional drug-like space and potentially have antimicrobial activity. And the other, particularly in the context of this conference, antibiotic research used to be collaborative globally, and how the original antibiotics were discovered was a global initiative. And possibly one of the best examples is vancomycin, isolated in Borneo from a soil sample. So isolated with soil samples in Borneo collected by a friend that he was in the war with. and isolated, sent to EC Cornfield at Eli Lilly, and then identified and advanced from there. And so one of the ideas of COED is, can we regenerate that collaborative global sharing without the barriers that are now in place with all the legal agreements that normally happen before you can even initiate collaboration. So COED was set up as a global collaborative initiative to seek new chemical diversity, accessing it from synthetic chemists anywhere in the world. And the way we incentivize it is we provide free screening against the bacteria of interest. So we test against 5 bacteria and 2 fungi. And the reason we're able to do that is we had significant strategic funding from the Wellcome Trust and support from the University of Queensland. So the pathogens that we tested against are 4 of the key gram-negative pathogens of concern. So Pseudomonas aeruginosa, Acinetobacter, Klebsiella, and E. coli. We do test against 1 gram-positive, methicillin-resistant Staph aureus, and we test against 2 fungi because the drug discovery pipeline for them is even worse than the antibacterials. And so we test against Candida albicans and Cryptococcus neoformans. We have tried to make sending in compounds as simple as possible. So we have our website, we have a button to click on, we have submission forms. You can send them in barcoded vials that we send you, or in plates, or just as loose vials, and we weigh out the compound. In terms of terms and conditions, we have a software license like a button that you can just click on. So we don't need a formal contract or MTA, but we do have a simplified version. Often the contributing university wants that. And basically the terms and conditions just say you have the right to send us a compound. It's not knowingly toxic, not radioactive, but the person contributing the compound retains all the rights to the compound. So we have no ownership over what they send us. And that is really critical for this open access initiative. So our workflow is compounds get sent to us, we plate them out, we do an initial screen at 32 micrograms per mil against our pathogens of interest. Anything that's a hit there that shows any signs of inhibition, we will then go into formal concentration dependent, so determining an MIC. Importantly, we will counter screen for cytotoxicity against mammalian cells, and we'll only advance something that shows at least some selectivity for the antibacterial or antifungal activity compared to the killing of a mammalian cell. We'll also do a quick QC to confirm the compound is what it's meant to be and is relatively pure. And then from a Wellcome Trust perspective, we have to do a novelty score because they don't want to see things that are similar to known antibiotics. If it's a hit, it goes into a larger panel where we validate it against other bacteria in the same class, including multi-drug resistant clinical isolates. We start looking at some of the drug-like properties. So is the activity inactivated if you add serum or lung surfactant? Is it stable to plasma within plasma? Is it stable to microsomes? How much protein binding is it? Does it cause red blood cell hemolysis? And then ideally, in collaboration with the person who submitted the compound, we'll do a resynthesis and do at least a little bit of early structure-activity relationship, because ideally, we don't want a singleton hit, that's the only compound that's active. We want to know there's some flexibility to do modifications because we want to be able to do a medicinal chemistry improvement. It's then up to the person who submitted the compound what to do with it. They can publish it, they can patent it, they can develop. Our only condition is that the structure and associated data has to end up in our publicly accessible database within 2 years, with an exemption if they are actually in the process of trying to develop it further. So we don't want to hinder the development of something that's promising, but we do want to have the data out there for people to be able to see what works and what doesn't. So as of last year, the initiative been running close to 10 years, 10-year anniversary this year, been highly successful in the uptake. So we've tested close to 350,000 compounds that have been submitted by over 300 research groups in 52 countries around the globe. And so there's a lot of interest and demand in people testing compounds for antimicrobial activity. And one of the things we'd like to think is we're helping to stimulate another generation of researchers to have some interest in looking at developing new antibiotics. It's led to over 130 research articles, poster presentations, grant applications, and even patent applications. So the data is being used in the real world to do stuff. we've had about 10,000, over 10,000 hits in the primary screening, of which about 3,000 of those are confirmed hits that are non-cytotoxic. And so for the 350,000 compounds, that hit rate is significantly higher than what you would get if you just looked at a traditional large pharma library. And so we think that the hypothesis that unusual chemical structures have greater activity is kind of validated. Our biggest problem is that you've got these 3,000 hits. How do you actually triage which ones are important to advance? And ideally, if we had a high throughput method of identifying mechanism of action, that would be the perfect way to do it, because you could figure out ones that are acting by unique mechanisms rather than just another membrane permeabilizer or something acting on a known existing target. In terms of the hit distribution, we had a surprising number of hits against the fungi. We weren't expecting that, given how difficult people report development of new antifungals is. Gram positive, we expected to have a lot more than gram negative, which is what we see. So about a third of the compounds that we've tested are now in a publicly accessible database. So we have the slope database where we have structures, and for each structure, we have the activity profile looking at the response against the different bacteria. And the unique thing about this data set, so 350,000 compounds tested under standardized conditions, and our data set contains all the negative results. And so this is something that often you don't find in the literature. It's the largest set of compounds that have been tested under standardized conditions against a range of different bacteria. So in terms of our initial hypothesis, do we have good chemical diversity? And so if you look at a PCA plot of the major physical chemical properties of the COAD library in blue, compared against the compounds within CHEMBL, which is a large chemical database of compounds with biological activity, we see we actually have pretty good diversity, and in some extent greater diversity than you see within CHEMBL. If you compare it against the commercial library in green, you see we have much greater diversity, which is fantastic. And if you compare it against natural products, you can see our distribution is focused more to the left, but we do have coverage over a similar scope as the natural products. So we think that our hypothesis that we're looking at greater chemical diversity has been validated. So just go through one case study. So within this COAD data set, we have this hidden wealth of information. And so we've got these novel compound classes, mentioned a large set of standardized data and negative results. About 5-6 years ago, we had a postdoc join our group who had an interest in organometallic compounds. He went trawling through the COAD database and found that we had just over 900 metal-containing compounds that covered 29 of the 49 possible transition metal elements, submitted by researchers in 17 different countries. If you looked at the breakdown of submitted compounds, the majority were ruthenium, closely followed by copper, iron, and cobalt. If you looked at the active compounds, ruthenium was still the top, but now you had the shift where you had the sort of noble metals rising to the top. So silver, palladium, platinum, and iridium complexes were the most active. And now if you looked at activity that were relatively non-toxic, now the platinum, silver, palladium rose to the top. And so these complexes had the best activity and were the least toxic to mammalian cells. And the thing that was most impressive was within all the metal compounds we tested, about 27% of them were active compared to about 2% of the traditional organic compounds. So you had an almost 10-fold higher hit rate for these metal complexes. And what was even more impressive was that carried through to the hits that were comparatively non-toxic. And this kind of went against the dogma that organometallic complexes have innate toxicity, which partly has arisen because the only drugs that are organometallic compounds are basically anti-cancer drugs. And so cisplatin is the prime example. So platinum complexes have the reputation of being highly, highly toxic. And this is where I think that this paper has had quite a lot of impact, is that it's helped to catch the wave and rejuvenate or enhance the focus on metal complexes as having antimicrobial activity. And the hit rate also carried through against gram negative. So our 0.1% hit rate in traditional compounds versus 1.5% for the metal complexes. So the type of structures, it's a whole new world of compounds, right? We're not talking about one type of structure. We're talking about multiple different metals that have all different types of complexes formed. So for example, the silver complexes are probably just loose associations of silver, which are probably releasing silver ion as the active species, versus the iridium and ruthenium complexes are highly stable three-dimensional structures where it's unlikely that metal is being released. It's probably intact, but potentially if there's redox activity going on, the metal complex itself might, because it's got this globular three-dimensional structure, maybe it's interacting with enzymes and receptors. We don't know, but potentially, all these different classes mean that metal complexes, potentially there are multiple different new antimicrobials within the overall group of metal complexes. So we published this in 2020 in Chemical Science. It got highlighted twice in Chemistry World, and I love the graphic they did for the second one. But sort of in terms of the impact, so that the paper has already been cited. It's now over getting up to 500 times. So it just highlights a lot of interest in development of metal complexes as antimicrobials. We did do a follow-up paper looking at specifically at the antifungal activity of the metal complexes. We now have about 100 more. And again, we had this dramatically higher hit rate of about 16% that were, 9% that were active with low toxicity versus 0.6% in the traditional compounds. And that got published in JAXA-EU in 2022. Our most recent initiative, we've now had a PhD student that focused on organoselenium compounds. Within our library of 300,000, we had about 233 organoseleniums. 77 of those were active, and 69 of those were active with selectivity for toxicity. And so in this case, the organoselenium compounds had a remarkable hit rate of 33% versus 2%. And even more amazingly, their toxicity rate was 87% non-toxic versus only 35% for the organic compounds. And in particular, they have some really interesting antifungal activity. So we're actively pursuing some ideas and hits that have come out of these organoselenium compounds. So in conclusion, from the co-ed analysis of metal-containing compounds, they have about a 10-fold higher hit rate compared to the traditional organic compounds. There's an incredibly large variety in the types of metals and the ligand systems that are involved. And so really, we're not talking about one new class of antibiotics. We're potentially talking about multiple new class of antibiotics within the organometallics. We've demonstrated through some programs that are focused on selected examples within the sets that small modifications to the ligand can have a large impact on the antimicrobial activity, which helps to show it's not just the metal center that's causing the activity, it's also the ligand surrounding the metal center. So that's something else to adjust. The other cool thing about metal centers, metal complexes, is that you can rapidly assemble very large libraries because you mix together different ligands, different metals, and you can build up a very large collection. And importantly, it has three-dimensional structure. So for those of you that know medicinal chemistry, there's been this so-called escape from flatland. So a lot of complexes now, or organo compounds that are made now, are very flat with structures, whereas things that are bioactive often have three-dimensional structures, which are much more difficult to make. And so synthetically, the metal complexes, you can get these very large three-dimensional structures very easily, which potentially gives them activity, useful activity, again, for receptors and enzyme inhibitors. The key takeaway is that the toxicity rate for our compounds that we identified is nearly identical to just traditional organic compounds. So they're not simply more toxic, but potentially are intrinsically more likely to be active against bacteria and fungi. There are some caveats, right? So relatively limited sample size. So we're talking about 1,000 compounds. The purity of the compounds that we tested is not guaranteed. So we assay as many as we can, but we don't assay everything. We do not know the identity of the active compound. As I mentioned, for some complexes, it's quite possible that the metal ion itself is being released, and that's having antimicrobial activity. But they're likely to have multiple and differing mechanisms of action depending on the compound and the complex. And what is very striking is that there is a ton of literature of metal complexes showing activity in vitro. There's almost no reports of them that have carried through a metal complex into in vivo studies. So there have been a few studies in Galleria models, which is a moth larvae model, but the number of studies that have taken a compound through into the sort of the gold standard mouse infection model could probably count on one hand. And so this is where the big gap is now. Organometallic complexes need to be taken through that traditional drug discovery pipeline. Also an issue in terms of, there's a lot of effort now trying to apply artificial intelligence, molecular modeling. The problem with organometallic complexes is that there's difficulty in the chemical representation of them in the electronic databases, both in the databases and modeling. Most of the programs are not set up to handle metal complexes and metal-centered compounds. And so there needs to be an effort to develop nomenclature that is able to accurately capture what the structure of organometallic complexes are. So moving on in the last few minutes into a completely different type of global initiative. So rather than focused on trying to identify novel antibiotics or novel chemical structures, this initiative is trying to build up a collection of highly resistant bacteria. So we're able to test our new antibiotics against clinically relevant strains. So this program was part of funding that we had from CARB-X to develop a new class of lipopeptides called the octapeptins. So the octapeptins are very similar to an approved antibiotic class called the polymyxins. Structurally, they look very similar. They have an interesting property of they retain activity against polymyxin-resistant bacteria. And polymyxin is kind of an antibiotic of last resort. It's very toxic, but is used to treat infections where every other antibiotic no longer works. The problem is polymyxin resistant can arise very quickly, even within a patient being treated with polymyxin. And the dose of polymyxin that is used therapeutically is very close or already exceeds the dose that starts causing nephrotoxicity. So you can't dose it at a higher level. So as part of our pitch to CARB-X for funding for our octapeptin development program, there's a component of CARB-X which encourages research focused on lower middle income countries. And so within our proposal, we pitched that we would be collecting polymyxin resistant isolates from lower middle income countries. And the reason for this is that this is where there are very high levels of polymyxin resistance. And you can almost look at these countries as sentinels for the levels of resistance that are eventually going to make their way into the countries like the USA and Australia. And so, for example, in countries like India and Pakistan, you can have more than 50% of the isolates are multi-drug resistant and have also developed polymyxin resistance. And so the idea is that by focusing on trying to collect polymyxin resistant strains, we would get these strains that also are resistant to most other antibiotics. One of the challenges in this is that we focused on a number of countries, particularly within Africa, where isolate collection, they'll have a patient goes into hospital, they'll identify what the bacteria is, but in general, unlike the pathology labs they have in Australia, the United States, and Europe, that isolate is not collected and stored. They just don't have the capacity or capability to do it. So you lose all that knowledge of what type of bacteria are causing the infection and what type of resistance they have. And so as part of the funding, we were paying each country that was, each research group that was collecting isolates for us, equivalent to what we would pay to ATCC to get a strain. So we're talking sort of between $300 to $500 US per strain that was being collected by these countries. And that allowed them to fund the researchers that were collecting the isolates and put in a little bit of infrastructure to be able to identify whether the isolate was resistant or not. For 4 collaborating centers, we also provided them with MinION sequencing technology and some training and technical support for them to be able to develop their own sequencing capacity. And the idea for this was trying to raise the level of research capabilities within some of these lower middle income countries. And so we ended up getting isolates from Asia, from Vietnam, Nepal, Pakistan, and Thailand, from Egypt, Nigeria, and Zambia, and a couple of different collection sites in Brazil, with a number of other collection sites identified, but we never managed to get through all the paperwork. And it varied greatly between countries, but trying to get isolates out of countries can be very challenging. And there are countries such as China and India, which basically do not allow the export of clinical isolates. And so looking at our isolate collection, the number of isolates we got out of each country, we also had a subset of isolates collected by a global agency that supports clinical trials for the major pharmaceutical companies that collects thousands of isolates from around the world every year. And we focused on some of the countries, lower middle income countries that we weren't covering, and a selection from their collection that appeared to have polymyxin resistance. So despite the fact that they're collecting tens of thousands of isolates, there were relatively few that met our criteria of being from LMICs and having polymyxin resistance. And so in terms of the species, we were focused on gram-negative, obviously. We had predominantly Klebsiella was one of the main ones, so this one in blue, and E. coli, with a few Acinetobacter and not many Pseudomonas. So we did have a reasonable variety of different species that came in. And so our process, we collected from 21 to 23. Isolates would come in from the collaborator, would make a stock preparation, would make some preparation directly, and would make some preparation on a polymyxin resistant plate to help select and maintain polymyxin resistance, which can drop off if you culture the bacteria without the presence of polymyxin. We specifically purchased a Vitek instrument, which is what is used in clinical pathology labs, so we could characterize, identify the bacteria, confirm the species identification of the bacteria, and get an MIC or susceptibility using the same methodology as most pathology labs do. But we also did a broth microdilution antibiogram against a panel of about 50 different antibiotics. So we have the Vitek result. We also have a traditional broth microdilution result. And then we did full Illumina sequencing of all the isolates, and then MinION sequencing of a subset of the isolates, which is still ongoing. So basically, the idea is having a large collection. So we've got approximately 800 isolates now that are fully phenotyped, fully genotyped, and we're putting together a publication that will describe those, with the idea that all the resistance genes combined with the phenotype will be very useful for applying for artificial intelligence predictions of, can you predict what the phenotype is from the genotype of the bacteria? So similar to the database we have for the chemical structures, we also have the database for the organisms, where we have the identity of the organism, its strain information, where it came from, the phenotype in terms of susceptibility, both from the Vitek and our broth microdilution, and then whether, according to UCAS guidelines or CLSI guidelines, it's considered resistant or not. And then we're able to track where those strains are, and then all the genotype information will be added in here as well. And so we've started doing some of the analysis, which is very interesting from being a chemist myself, trying to get into all the genomics of bacteria. But we see this large diversity. For example, with Klebsiella, we've got a large amount of diversity. We've ranked by whether they're polymyxin resistant or susceptible, and then start looking at all the different genes within them and their antibiotic resistance genes that are present or not. And so there's a wealth of data in here that basically will skim the surface, but then we'll put it on a public database for other people to have who know more than us to be able to have a look at and play through all the data as well. And basically our overall goal is to basically set up an antimicrobial knowledge base where we've got our chemical structures and the biological activity. We've got these microbial strains and intersect with the biological activities. And all this becomes publicly accessible database. So you can start applying artificial intelligence to our chemical structures to be able to predict what an antibiotic looks like. You can apply it to the microbial strains with the genetic sequencing, trying to develop better predictive assays for identifying what bacteria might be resistant just based on their sequence to develop rapid diagnostics. But I think what I'd really like to emphasize is that none of this work would be possible without the global collaborations that we've established. And so we're very much standing on the shoulders of everyone else that has contributed to this work in order to get these results. And that is going forward, I think for you guys as well, very, very important in terms of developing new antibiotics is hard. Antimicrobial resistance is a global challenge. We need to be working together and not in silos, and making use of the expertise of everyone around you for the greater good. And that's, if you have one take-home message from this talk, that's what I'd like it to be. It just goes to thank the team of workers that has actually done all this work. We've got a very multidisciplinary group. We've got a bunch of synthetic chemists, we've got a bunch of microbiologists, and then we've got people that kind of bridge the gap in between, and that's been critical for doing what we do. Also thank the funders, so had Wellcome Trust funding, Carbex funding. The subset of funding that funded that isolate collection actually came out of GAMRIF, which is part of Carbex. We've had some funding from GARD-P to do some screening, and then the traditional funding of ARC and HMRC, and we had a diagnostics program from DMTC. So thank you for listening to me, and I'd be happy to answer any questions.
Thank you very much, Mark, for that extremely engaging talk. It's much appreciated. So now we move on to our Q&A part after our keynote session. So for all our participants, at the bottom right-hand corner, you should have the Q&A function in Zoom. Just put your question into there, and Mark will answer it as we go, and until 9:15 till our next session starts. So please, by all means, Q&A function is open. While we're waiting for people to put their questions in, I actually might ask one question myself. So, Mark, you mentioned that there's a drastic downtick of new antibiotics being approved by the FDA. And yes, there is the lens of it simply being a redevelopment of old antibiotic drug classes. Is there a reason for this reduced approval?
Yeah, I mean, it's just a number of compounds, new drugs that are being developed and submitted for approval. And it goes across all the regulatory agents. So the FDA is the easiest one to track because pretty much every antibiotic goes through there so they can access the US market. But it's similar with the European Medical Association. There are a few antibiotics that have been approved in China and India that are not available, or even Japan, that are not available in other countries yet. And going forward, that actually might become more common because there is quite a lot of research going on in both China and India. Partly because they have a greater demand. There's a lot more antimicrobial resistance there. A lot of people are dying from infections where no current antibiotic works anymore. So the future may be that antibiotics are largely originating from some of those countries rather than from the traditional US.
Great. All right. We have a couple of questions from chat. So question one from Samuel. Are there any known mechanisms by which bacteria develop resistance to metal-based antibiotics?
Yeah, so we don't know yet. The groups that are focused on organometallic complexes, so some of them have tried doing mechanism of action studies. It looks like reactive oxygen species are involved, but that pathway tends to be involved with every antibiotic anyway. So whether it's a primary cause or a secondary effect, it's not clear. I mean, it would make sense with metal complexes with the redox activity that reactive oxygen species could be one of the main mechanisms. But again, it's going to depend on every different complex is going to act differently.
Great. Thank you. We have a question from Anton, who is asking, what are your thoughts about antimicrobials targeting virulence mechanisms?
Yeah, look, that's a great question, Anton. Thank you. There definitely is room for them. I guess so if you're looking specifically at antivirulants that have no innate antibacterial killing activity, there's certainly potential to reduce the pathogenicity of an infection and then potentially allow the body to naturally clear it or for antibiotics to work more effectively. but reducing the toxic effects. One of the things that's always spruiked as an advantage of antivirulence strategies is that they would not lead to antibiotic resistance. I strongly disagree with that proposition, because I think anything that affects the fitness of a bacteria, the bacteria are going to find ways to overcome it. So I think even if you just had a pure antivirulence strategy, eventually you'd find bacteria that manage to escape that and regenerate their virulence factors. So I think it's definitely got its place in the development of new approaches towards therapeutics. And the other thing that I've completely ignored in my talk are non-antibiotic approaches. So obviously there's a huge amount of interest in phage therapy, for instance, and other less traditional approaches. And I think they all have their role to play. Specifically on phage, I think there's great potential for it in selected cases, but I don't see how it could become a general therapeutic replacement for antibiotics. Maybe working in synergy with antibiotics would be the best approach for that.
Great. Our next question is from Hasana, and she's asking, what is your idea about using the chelation strategy and nanocarrier systems to enhance metal complex efficiency?
Yeah, look, I think so two different, I think there are kind of two different questions within there. So in terms of chelation strategy, if you're talking about using siderophores to help get compounds into bacteria, that's something that has potential. We've got some active research programs into looking at that. For nanocarrier systems, I think it's probably too early to start looking into that specifically for metal complexes, because I think we need to identify whether metal complexes satisfy the criteria as potential drugs first, and then look at potential ways of improving their delivery. So I think that getting the fundamental of identifying this is a really promising compound, it's got the right properties, it's not being degraded quickly, then can we deliver it more effectively to get it where it needs to be.
Another question we have from Pradeep, which is, do these purely organic drugs or organometallics also have some role, I'm guessing, in the resistance mechanisms or efficacy?
Yeah, Pradeep, if you're able to elaborate on that, what role in what?
Yeah. While we wait for Pradeep to sort of type a reply in, I'd like to get your take on the new, the fact that there was a E. coli vaccine for antimicrobial resistance E. coli that was, I think, from a Johnson & Johnson related company that did not proceed through phase 3 trials.
Yeah, I think I think vaccines are a potentially incredible effective strategy at reducing the need for antimicrobials, and obviously, the meningitis vaccine, the pneumonia vaccine have been incredibly effective at reducing the need for antibiotics and reducing deaths caused by those infections. I think the failure of the E. coli and Group B strep vaccines, there's been a lot of effort into trying to develop antibacterial vaccines, and it's very, very difficult to be successful. Partly because there are so many different variations of bacteria that you need to try covering, and there's no universal epitope that will target them all. And it just, I think it highlights how lucky we were with COVID and how quickly we were able to develop multiple different vaccines. Traditionally, vaccines for other things have taken much longer and have not been successful. And we still don't have an HIV vaccine example. And bacterial ones are even worse and more difficult to develop. So I think it's going to be, one arm of the strategy of combating resistant infections in the future. And I think eventually we'll get there, but it's just gonna take time.
Thank you, Mark. Last question from Anton. Oh, metallo-beta-lactamases, which are zinc-dependent, remain an Achilles heel of treatment of patients with gram-negative infections. Thoughts of new inhibitors based on your metallo-compound work?
Yeah, interesting question. We haven't specifically looked at whether any of them have activity against the metallo-beta-lactamases. Maybe that's an opportunity to get some isolated enzymes and screen our library against them to see if there is any selective activity. But yeah, the other approach people have been trying to use is zinc starvation, of getting rid of the zinc and taking it out of the enzymes as well. But again, that's going along the chelation type of strategy. But it is challenging because you need zinc in your body. So if you deplete all the zinc, your body's not so happy.
Right. Excellent. So there's